Hello, and thanks for joining me again for our May edition of The Researchers Behind the Images for our 2025 calendar. There is a downloadable version available on our website if you haven't managed to pick up a hard copy. Again, my name is Jess, your representative from the Antibody Society, and today I will be joined by our May image shown behind me.
So welcome back to our May edition. I am joined here by Rodrigo from Amsterdam UMC, where he works on methodology and personalized medicine. So I know that you're a late PhD student, am I correct there? Can you tell us a bit about how your project's been going?
I think it's going well, but yeah, I'm a late PhD student. So I started working here as a PhD student as part of the Marie Curie ITN Cosmic, where we combined computational approaches and experimental approaches. So that was really nice, modeling and bench lab. And I'm continuing my PhD, working also on computational approaches to understand better antibodies, TCRs, having fun.
Brilliant. Well, I know that a lot of places are moving towards incorporating these sort of computational models as part of how we generate our monoclonal antibodies, which is highly relevant to the Antibody Society. So I'm really looking forward to learning some more. Now, the image that you generated is 100% computational, and it is absolutely stunning, as you can see behind me. We're very happy to have some diversity in our calendar, and you're going to teach us how you actually generated images like this, am I right?
Yes, and it was also quite nice to think how to prepare this type of image. Yeah. Yes. So the name of the image is Clonal Fireworks, and I think it's pretty straightforward.
Yeah.
So I'm Rodrigo Garcia, as you said, part of the Amsterdam UMC, and let's talk about fireworks. For me, working with antibodies and BCRs and clonal expansion, everything is really interesting. So I thought of a nice way to showcase this type of population dynamics, and I'm going to talk about this. So first of all, the data that it's used to generate this image, it's a very well-known data set from Laserson and Vignold from more than 10 years ago. It's human BCR heavy chain repertoire 7 days after flu vaccination. So you can see that some clones have been expanded, others haven't been expanded. And this is also part of the Alakazam package, which is a very used R package. So, yeah.
So how many clones are there found within this data set? Do you remember how many are represented in the image?
I don't remember, but I guess a couple hundred. It's not that much.
Yeah, so is that normal number to expect from this sort of data set?
No, this is a small subset.
Okay.
So it's a downsample of the original repertoire.
Brilliant.
And well, what I can say here is that we have clones that are non-singleton. That means that they have more than one subclone and they are expanded, and others are singletons which are not expanded. And the singletons are represented as a dot, and the other ones as a radial plot. You can see the number of leaves in this radial plot represents the number of subclones that are within that clone. So it gives an idea. And the bigger the radial plot, the higher it is located in the plot.
No, that's amazing. So is there an average number of sort of your radial like single clones that are coming off your central nodes there?
Yes, but I don't know what's the average number, and probably it's probably the distribution is not even normal. So
Yeah, no, that's brilliant. It looks quite variable, but yeah, it makes a beautiful visual representation. I think it's very useful to help people think of it in a very different way to what you normally would. It's interesting new graph that we could come up with.
yeah, I think that many times when you look at the bar plots or box plots or whatever you use for getting an idea of the distribution of the clones, you never get to see this visually. So that's why I wanted to explore this way of visualization. And I also added a new dimension, which is the color, which is based on the VDJ recombination. So each clone is color based on its VDJ usage. But the thing is that similar colors indicate clones that have similar VD and or J genes, so
Yeah.
that they belong to the same family or same thing. And for this, I used you know the hexadecimal colors, right?
Yes, yes.
So I did a small trick. So colors are a mixture of red, green, and blue. Depending on the mixture, you get a different color. And in this particular case, I assigned specific ranges of red colors to certain V families, green colors to D families, and blue colors to V gene families. So V genes that belong to the same family will have similar shades of red colors than when compared to other V genes that belong to a different family. So, for example,
Okay.
the most abundant clone, it's a mixture of these combinations of red, green, and blue, and that's what gives this nice pinkish color.
That's a really unique way of thinking about it. I did not expect it to have this level of depth into which clone was which. That's amazing. It gives me a whole new appreciation for your image, actually.
Thanks. To be honest, there are a lot of plots that represent different VDJ usages, and I thought that they really don't. Sometimes they are a bit misleading because you have V genes that are very similar between them and they are colored in a very different way. So I thought of a way of making this more interpretable. And if you look at the image, the clones that are expanded are clones with a very similar VDJ usage. So there is something in this VDJ usage that allows them to be expanded. And in the background, you can see singleton clones with different colors, which belongs to very different VDJ families. And in the lower part, you can see very expanded, very lowly expanded clones. Some of them belongs to, let's say they have other colors because they have another VDJ usage, but still they don't get to be very expanded.
Okay. Yeah, so is it normal that so most of them seem to be more on the red spectrum. Is it normal that they should be highly related in the ones that are expanded, or would you have a different subset where they're mostly green or mostly blue or things like that?
Probably if I apply this to different samples, the results will be different. In this particular case, it seems that there was some predisposition by these particular VDJ combinations to be expanded, I guess.
Yeah, it's predisposed by the biology behind it, which is a very good thing.
And I also tried different things over this image, but I didn't include them finally. So, for example, I tried to include also the subclonal abundance. Some dots on the radial plots will be bigger if these subclones were more abundant. I also tried to include the number of mutations in the plot, but that's a lot of information to represent in one single plot.
So you could have different versions of this highlighting these different types of things, but all overlay that you could visualize each thing separately, because we can't take in that much information at once, as you said.
So I kept it simple, but still there are a lot of things that you can do.
Yeah, I wouldn't say this is exactly simple, but no, it's beautiful, interesting. So you were saying lineage trees as well.
I tried to represent the radial plot as a lineage tree, but still this is fancier. This is a firework per se, so I kept it as this. And a lot of the things that I developed for this image, I reused it for another program after this. So this is something that I have been working on for the last couple of years. It's called AV Solution, and it's an interactive tool for analysis, exploration, and comparison of variable region sequence features of B cells and T cell receptors measured with AirSec. So it's a long name, but I will explain it very briefly. And the thing is that this starts with, of course, everything starts with a story, and I don't know. I'm really excited about AirSec data. I think it has a lot of potential. It's very complex, but it's fun. So there are lots of data sets. There is a lot of information contained within these data sets. It's multi-level, interconnected. It has a lot of layers, and there are so many scenarios that when I started tackling some troubles or some type of biological issues, I thought on a transversal way to explore everything. So I prepared a program that is scalable. You can add a lot of samples to it. It also has a lot of calculates a lot of features per every sequence. It's interactive, and it's also very flexible, so you can adapt it to the different analysis that you want to do.
Brilliant.
And this is the solution that I developed, which is called AV Solution. It's scalable, it's interactive, and it's flexible. It's an R-Shiny application where you, well, it's easy. You upload your data, you set up the project information. You give some information on how to reconstruct the gemline, because that's very important. You decide if you want to use the digging information or not, if there is a Persian framework region 1 or Persian framework region 4, because sometimes you don't sequence everything at all. And then you calculate features for every sequence. And these sequences are calculated for the whole variable region sequence. And for each region, framework region 1, framework region 2, or CDR1, CDR2, etc. So the sequences are a lot, as you can see. Different categories, physicochemical. They are related to somatic hypermutation or to amino acid properties, sequence motif and composition, amino acid descriptor sets. And you calculate these sequences for the gemline, for the infrared gemline sequence, and for the repertoire. And you can see also how this, in this case, the TCR, it has changed from the gemline to the current sequence. So
brilliant.
This is nice, and you can explore many things, the number of glycosylation sites, the number of insertion deletions, but also charge, bulkiness, hydrophobicity. The thing is that you get a lot of features, and in some scenarios, these features may be of interest. And the next steps, you can explore this data in different levels. There is one level, which is the sequence exploration level. You can filter data by sample, group, VJ uses, region, feature type. You can sample, normalize. You can compare sequence group. For example, in this plot at the left, we have separated dominant and non-dominant clones that have a particular VJ usage in this patient. And this is a PCA that represents all of these features that we have calculated. It represents the significant features that separate these two. You can also go further. You can explore these features in between groups. And you can also compare these features against the gemline references and test different groupings. In this image, you can see two violin plots between a subset of TCRs that have been exposed to a particular antigen. So some of them have changed from day 0 to day 10. They have been expanded. And you can see that the TCRs that have been expanded at day 10, they have a higher hydrophobic moment in the CDR3 compared to the baseline.
Okay. No, that's brilliant. So they also have all of your TCR information in there. Can you tell like what T cell subsets they are and look at closely at those sort of things, or we just know they're T cells?
To be honest, I'm very focused at the app programming level, so I don't have it in mind. So I'm testing this in different data sets that they are published. It's true that I haven't put the reference here, but well, I'm already working in the paper.
That will all be in the paper. That's absolutely fine. So what do you look for when you're selecting sort of your data sets to put into this? Because we'll have a lot of younger early career researchers who are interested in expanding into these sort of fields. So what do you look at as someone who has a lot of experience as to how you select the data sets as being good quality and things like that to include?
Well, I mean, basically, you need to think about the biological question that you want to address, and understand the data sets per se, to see if there are some limitations, to see if you can combine different data sets from different sources. because sometimes it's not that feasible, or you need to downsample to standardize the comparison. And I will look for data sets with a good sequencing depth, good quality, good biological metadata.
Yes.
And so something that you can trust and explore, and you are familiar with the subject that you want to explore. This comes from my experience when I have explored AirSec data. So there is a lot of things that are intertwined, and I wanted to find a way to explore this in an easy way and to test different hypotheses quickly. This type of application, it's interactive, so you can say, I want to check this subset, and then you see what's the effect on this subset in not only in between sequences, but also between clones. I will show it now. So this application also allows you to explore clones, and you can see, you can test different clonal definitions, you can set dominant thresholds, you can analyze how the frequencies are distributed, and you can see shared clones in between samples. So in this particular plot at the left, you can see that, for example, I don't know if you see my arrow, but
yes, we see it.
These clones, they are over a threshold, over a dominant threshold. So these clones are, they could be considered dominant.
Okay.
And these are the sequences that are between day 0 and that are shared, TCR sequences shared between day 0, present in day 0 and in day 10. And in the next plot, you can see which of these sequences are shared, which of these clones are shared between day 0 and day 10.
That was what I was going to ask.
You can click here, and you will select the clones in the previous plot, and then you could do more analysis, and you could, for example, see what's the shared characteristic between these shared clones and the non-shared clones. The difference between these groups. So there are many things that you can try in this application, and that's what I like, and that's what I wanted to explore here. So you can work in these different levels, depending on the sequence that you want to explore. You will see different. You will, and the grouping you do of these sequences, you will see different features, and you can do different work at the clone level and at the sequence level. It's nice. And finally, it's also something that I believe it's very important. It's to be able to export the results and to be reproducible, especially when you work at on such a complex level. So in our lab, we published last year a paper about computational reproducibility. This is a transversal method of reproducibility in computational analysis. So I follow these guidelines, and I implement. The user can include some of his or her information and notes along this analysis. So it will be kept for when you share the report. That information will be kept. The report will keep the input parameters. It will have the code to reproduce the results, the system and run information, and more characteristics. So if you share the output, any person can reproduce it for sure.
Oh, brilliant!
For sure, because it's very complex. But we did a lot of work into this.
Yeah, no, well, that's hugely important. You have to be able to trust the data that's coming out, especially when you didn't write the code. Sometimes it's really hard to understand what exactly they did. So it's nice to see that that is at the center of what you're doing as well. No, that's brilliant. So in coming forward, when this does become available, what sort of applications do you see this working, being useful for? For example, people you would think within the Antibody Society would find this sort of tool useful.
I think it would be good to get an idea about what's the difference between two conditions or between two groups of some things. And it's the answer is never perfect, but only knowing that, for example, maybe there is a difference in some characteristics in the CDR3 will give you some starting point to do bench work. Also, we produce a lot of data for every sequence, so this data could be reused for more complex machine learning applications. This is an image of the application, the way it works.
Yeah.
So you can test different things. You can also click the sequences that you believe are more interesting. You can export it. You can keep track of everything. And
that's brilliant.
yeah, you know, this is the teams I work with, and they are great. And
Cool.
I also wanted to say thank you to the Antibody Society. This type of initiatives are really good, and I
Oh,
think
thank you.
it's really nice that you do this and promote people and promote even the Antibody Society itself, which is what it should be. And this is finally the final presentation. So
Yeah.
any idea, comments are always absolutely welcome.
Thank you all very much for joining me once again as part of this series. We greatly appreciate all the interaction we've been having with our members through this initiative. On behalf of the Antibody Society, I hope to see you again very soon in the future.